everyone, welcome back to my channel. This is Soumya from Sri Venkateswara College of Engineering, and today we'll be discussing about the properties and the functions of enzymes that are also called the catalysts of life. Enzymes are the proteins that act as a catalyst, speeding up chemical reactions in living organisms. They bind substances, lower the activation energy required, and facilitate the conversion of reactants into products. Enzymes are crucial for all biological processes, from digestion to our cellular respiration. Let us go through the enzyme structure and their functions. Structure of enzymes have a unique 3D structure with an active site that binds to specific substrates. This structure is determined by the sequence of amino acids that make up the enzyme. Uh, Functions of enzyme. Enzymes work by lowering the activation energy required for a reaction, allowing it to occur more easily. They do this by stabilizing the transition state of the reaction. Specificity of enzymes. Enzymes are highly specific, meaning they only catalyze a particular reaction with a particular substrate. This is specified that is due to the shape and property of the active site. Now let us look into the factors that are affecting the enzymic activity, which are temperature and potential of hydrogen and substrate concentration and enzymic concentration. Work at best usually around 37 degrees Celsius, that is our body temperature, and temperatures or above or below this can decrease the enzymic activity. Potential of hydrogen, that is pH. Enzyme functions at its best at a specific pH level, which varies depending on the enzymic activity. At extreme pH can denature and inactive enzymes. Substrate concentration. Enzymic activity increases with the substrate concentration up to a point, after which it levels off due to the enzymic saturation. Enzymic concentration, that is increasing the amount of enzyme present will increase the rate of reaction, and more active sites will be available. Enzyme kinetics. Enzyme kinetics is a field of study that attempts to classify enzymes by the sequence of reactions necessary to form a given product and to understand which enzymatic steps of the process are rate limiting. The principles of enzyme kinetics are substrate binding, catalysis, and product release. Substrate binding, when the enzyme binds to the substrate to form the enzyme substrate complex. And catalysis is when the enzyme catalyzes the reaction, converting the substrate into the product. And product release is the when enzyme releases the product, returning to its original state and ready to catalyze another reaction. Enzyme regulation and inhibition. Regulation. Enzyme can be regulated through an allosteric interactions, covalent modification, or control of gene expressions to adjust their activity as needed. Competitive inhibition. Competitive inhibitors bind to the active site, preventing the substrate from the binding and reducing enzymic activity. Non-competitive inhibition. Non-competitive inhibitors bind to a different site of the enzyme, changing its shape and reducing its activity. Non-competitive inhibition binds to a the inhibitor binds to a secondary site on the enzymes, and this changes the shape of the active site and prevents the substrate from the binding. Feedback inhibition. This occurs when the end product of the pathway inhibits an enzyme earlier in the pathway, regulating all overall processes. Now, let us understand how enzymes perform in metabolic pathways. Step 1 is when enzymes catalyze the first reaction in the metabolic pathway, that is, converting one substrate into an intermediate product. And step 2 is when the enzyme in the pathway that converts the intermediate into the next product in the sequence. Step 3 is when the cascade of the enzyme catalyzes catalyzed reaction continues and ultimately producing the desired end product of the pathway. Now let us understand some therapeutic applications of the enzymes that are digestive enzymes that is used to treat the conditions like lactose intolerance and pancreatic insufficiency. Clothing enzymes used to break up blood clots and treat conditions like deep vein thrombosis. Anti-cancer enzymes that some enzymes can selectively target and destroy cancer cells. Industrial enzymes that are used in laundry detergents and biofuels and other industrial processes. I hope you all enjoyed the video and thank you for staying tuned.